All right, so it's going to start after all that lovely explanation. All right. So in the last question, yep, for test 1, is that CAMP affects a lot of things. Why do we do that? And this is something we actually touched on later on in the semester as well, is that you can have this, what's called, I even have it in the question, cascade effect. CAMP goes and turns on one molecule. This can go and turn on other molecules. This can go turn on other molecules as well. So by just releasing or having a small change in one molecule, you can affect a large change as long as the signal is being amplified. And we learned later on, now, if you have other signals that can work with CAMP, you can actually fine tune your system where I have multiple signals happening at once that are turning on different pathways and mainly modulating each other. And it's a way, it's a more or less kind of like a language inside the cell. It's where based on what sentences, or in this case, what substrates molecules I'm seeing and what quantity I'm seeing, I can start to modulate my behavior. So maybe with all the signals that are out there, I turn down protein production on average 20%. And so that's the way we can use like a dial by having these multiple signals to start regulating behavior. At this point for test 1, we didn't really talk about that. So for the test 1 answer itself, it's a way that we can have a small change in something, in this case, CAMP. And that small change leads to a big result because we keep turning on multiple things. And I think in class, there were example I gave, CAMP turns on like 5 proteins. These 5 proteins turn on other 5 proteins. Those other 5 proteins turn on another 5 proteins. So just by CAMP, I had an increase of like 125, something like that. But that was the last question on test 1. So anything in general about that before we move on to test 2? All right, test 2. Here I actually have the key, so now I don't have to think on my feet. Perfect. Test 2, a person is born without a mutation that causes their cells not to produce complex 1. Can you still produce any energy at all? So I think this part tricked some of you up, because some of you said, no, if you have complex 1, you can't produce energy. Well, complex 2 will still produce coenzyme Q. It's not going to be as good as complex 1, but you still have some energy being produced. So if you're born without complex 1, you're probably not going to live, but you could probably still produce some energy. So technically, yes, complex 2 can still produce Q. Why is oxygen necessary in aerobic cellular respiration? Oxygen is our main electron acceptor. It's the last electron acceptor, rather. So if you stop having oxygen there, for example, you stop breathing, well, you don't have anything to deliver oxygens to in complex 4 in your ATP synthase, rather, at the end. If that stops, the whole chain is going to stop. So you're going to stop all the complexes. You're going to stop the citric acid cycle. You're going to more or less stop glycolysis, although that can shoot off to anaerobic. And you're going to overall stop your whole ATP synthesis process. And so to avoid that, you need to breathe oxygen to keep your electrons flowing. Oh, don't want to go down that far. Oxidase phosphorylation, we've talked about many electron carriers. For each of the following, please describe where they're found and their roles in oxidase phosphorylation. All right, so I remember in this one, I guess my wording was a little vague. what I meant when I said where they're found, I meant like, what complexes do they come from and like where they travel. Some of you answered that question in a way I wasn't intending, but it was technically right. So I still gave you the points. But here's what I was looking for. So Cytochrome C, this is going to be your electron carrier that bounces out of complex 3. And it actually travels through the inner membrane space and then goes to complex 4. Iron sulfur complexes. So I believe this was the question where people said they're found in the inner membrane. And yeah, technically that's correct. So what I was looking for was what complexes were they in. So I probably should have said that. They are in complexes 1 and 2. That, if you remember, when we are passing electrons, we can only generally do it 1 electron at a time. And so our iron sulfur complexes help to form a ladder down complex 1 and 2 that we are handing off 1 electron at a time to. So if NADH, FADH2 come in, we give our electrons away, then they will ladder down the complex until they end up at coenzyme Q. And coenzyme Q, ubiquinone, that's from complex 1 and 2 again. And you'll get electrons from 1 or 2. You'll go in the inner membrane space and give electrons to complex 3. Questions about that so far? All right, carnitine shuttle system. So carnitine shuttle system, that is what is allowing us to bring fatty acids into the mitochondria. I don't think I asked you a question on that. So another thing you can cross off your list of having to review. Fatty acids, though, breaking down and building up, you're going to want to review that. There are some questions that get into that process. We did spend a bit of time with it. So I have a fatty acid coenzyme A molecule, 10 carbons. I label my carbonyl carbon with radioactive C14. If I'm doing beta oxidation, how many rounds do I have to do to get a label acetyl-CoA? That's just 1. You're going to work, the way it works is that you cut 2 carbons off and you always start from the carbonyl spot. So 2, and if this is radioactive labeled, that's going to be the first thing that flies off. But it goes in 2, 2, 2, 2, 2, 2, 2. How many hydrogen atoms does it take to make 1 molecule of ATP? Basically, it takes 3 hydrogen molecules to spin the whole ATP synthase, and 1 complete spin will give you an ATP. So it's just 3 atoms to make 1 molecule of ATP. APO. Anytime you see the word APO, and for some reason, I did not learn this until I was like a 3rd or 4th year grad student. It never clicked. APO just means you have a protein complex. Well, let's go. Let me step back. If you don't have APO in front of the word, that means you have everything you need to make an enzyme or a protein. So you have all your cofactors. In this case, you have any lipids. APO means you don't have all that stuff. You just have your protein. So anytime you see the word APO, that means this is just the protein part of your complex. All the other stuff you need to make your enzyme work, you're missing. You just have bare protein. ATP synthase has 3 conformations: open, loose, and tight. What is there? Open, nothing's there. Loose, ADP and phosphate. Tight, you smash those 2 things together and you have ATP. There was, I believe, some debate I had with a couple of you where I believe that, if I remember right, is that the open conformation could technically have ATP in it because that's where you release it. And I'm going to rule, we're going to call the open conformation nothing. I'm going to rule that when you go from tight to open, that's when you're releasing the ATP. And so when you're in the completely open conformation, you no longer have anything bound. When you're in the loose, that's when you bring your ATP and phosphate in. And then when you're tight, that's when you smash your ADP and phosphate together to make your complete ATP molecule. The Q cycle, here's another bonus for you. Q cycle is great, but we have a ton of cycles. And so don't worry about memorizing the Q cycle for our final.
Weak. No, I'm just playing. I'm just playing.
You already spent time looking at the Q cycle?
No, it's just one of those things I remember.
Oh, well, that's good. I'm not going to test you on it, but I'm glad to hear that. All right, so what is the extra product resulting from beta oxidation of a saturated fatty acid with 19 carbons as compared to a fatty acid with 18? So if you have an odd number of carbons, you're always going to end up with propanol coenzyme A. That's what you always get. So I could put any odd number carbon here, and you're going to end up with propanol. Okay, whoa, there we go. What's the logic of citrate being an inhibitor of glycolysis? Okay, so this is another one of our big picture questions. Citrate is made from the citric acid cycle. If you're running the citric acid cycle a lot, you'll get a lot of citrate. And you're running the citric acid cycle a lot, that means you have a lot of ATP. Citrate is one of those rare molecules that can be in the mitochondria and also in the cytoplasm. Glycolysis happens in the cytoplasm. So if you have a ton of citrate leaving the mitochondria, that means you must have a ton of citrate overall. So much so that the citrate levels build up in the cytoplasm enough to inhibit glycolysis. And the logic is, I have a lot of ATP, stop running glycolysis. If you have a ton of ATP, we don't need to run glycolysis anymore. Start storing that glucose, or maybe run the phosphate pathway, the shunt pathway, or do something else with our resources, because right now we do not need them. So that's your big picture logic question on citrate. Citric acid cycle, nothing much to say here that I didn't say for glycolysis, but since I didn't record glycolysis, I guess I'll just repeat it. I'm not going to ask you to fill out a chart like this, but you should know basically for all cycles, why are some reactions spontaneous or are reversible or non-reversible? So for example, we have some non-reversible reactions here. Why do we have some reactions that are reversible, non-reversible? Non-reversible is usually a stop point, or not a stop point. It's usually a point for regulation. Also, for reactions like taking malate to oxaloacetate is usually not a favorable reaction. Now I'm thinking of citrate synthase. Citrate synthase usually, no, it's malate dehydrogenase. Sorry. So citric acid cycle, we have reactions that are large positive. Why can they run? Because you are putting them next to reactions that have delta G very negative. And that reaction could happen actually not right next to it. So you can have like positive, 0, 0, and then a big drop off into negative. So positive, 0, 0, big drop off. And this drop off down here is going to pull the whole thing forward. So as long as you keep your products at a low concentration, you can have reactions that are usually non-spontaneous on paper, be spontaneous. So conversion of pyruvate to acetyl-CoA, what cofactors do you need? TPP, lipoic acid, and NAD+. And since we have talked a lot about pyruvate dehydrogenase, this is one that I will not cross off. So this is totally fair game. So describe the reactions between enzyme 2 and 3. So I just put the whole pathway up here. So pyruvate, you knock off a carbon dioxide, you attach that to TPP. From there, you take your lipoamide and you put your acyl group there, and you can bring in coenzyme A, chop off your acyl group, make acetyl-CoA. So that was, so I want enzyme 2 and 3. Okay, so 2 and 3 is coming up now, where we have our sulfurs, we have broke our disulfide bond. Well, I want to take another acyl group, so I need to reform this disulfide bond. The way I do that is I break my disulfide bond in E3 to reform my disulfide bond in E2. Well, I need to reform my disulfide bond in E3 as well. So NAD plus will come up, reform that disulfide bond, and make NADH. That's your refresher and pyruvate dehydrogenase. Glycerol phosphate shuttle. Mitochondria is, especially the inner mitochondria matrix, you don't let a lot of stuff in. So we have these weird shuttles that will work with the molecules we can let in, and as a side reaction, will effectively transfer one molecule from the cytosol to the mitochondria. So for here, for the glycerol phosphate shuttle, we cannot get NADH in. So what we're going to do is we're going to take an NADH in the cytoplasm. We're going to change that to NAD plus. And the glycerol phosphate shuttle will bring those electrons, nectar, hydrogen into the mitochondria and take an NAD plus there and reform it into NADH. So that's what the glycerol phosphate shuttle is doing. It is effectively transporting NADH into the mitochondria for us without actually physically moving molecules. We're just moving hydrogens and electrons.
So the hydrogen isn't attached to anything. It's literally just a loose hydrogen?
No, it's attached to a molecule that I have to look up because I don't remember off the top. Yeah, I don't remember off the top of my head.
Okay. It's just like going from oxaloacetate to malate, putting two hydrogens on there.
Yeah, it's the same exact idea.
Okay.
For the glycerol phosphate shuttle, where you attach two hydrogens, you attach those electrons, and all you're going to do is chop those hydrogens and electrons to an NAD plus in the mitochondria to make NADH.
Okay.
So ATP synthase can run backwards. It would be super bad if it did. So why doesn't it? That's because we have an inhibitor, IF1. IF1 is basically a stick. And this stick can come in two forms. One is the tetramer form, which has 4 copies of itself. And in this form, it doesn't really do anything. It kind of just floats around and doesn't interact with anything. However, if the pH starts to change, if the pH starts to become normalized from the inner and outer mitochondria area, the matrices, what will happen is that this tetramer will break off into dimers. And in the dimer form, IF1 is now active. So it'll go to ATPase. And the reason I said it's a stick is because if you remember, the ATPase is like a giant rotor. It just kind of turns. IF1 kind of just sticks itself in there to physically prevent this ATPase from moving at all. Because what would happen is if my normal rotation is clockwise, what will happen if the proton gradient isn't kept is I can accidentally start running counterclockwise, and it's going to use ATP to do that, which is super bad. So we just put our IF1 in there and physically stop any rotation from happening. And the only way we're going to get that stick out of there is if the pH starts to renormalize, because in different pH conditions, IF1 is either a tetramer, so it'll want to form a tetramer, or it's a dimer. And that's completely controlled by protonation and deprotonation of amino acids on IF1. Okay, one complete cycle of beta oxidation. So if you all remember, double bond, OH, C double bond O, cut. So hopefully that is ingrained into your mind forever and ever and ever, because I've probably said it like 50 times now, but here we have our C double bond O, carbon alpha and carbon beta, and we're going to form a double bond between alpha and beta. And I add an OH on the beta. Beta becomes C double bond O. And what used to be our carbon alpha beta bond, coenzyme A is going to come in there, cut that, get an acetyl coenzyme A out, and a fatty acid coenzyme A. And then lastly, on test 2, before the world ended, we were talking about fatty acids. So triacylglycerols, we want to digest those. So what is the cholesterol derivative? That was your hint for that. That will do that for us. Bioacids are derived from cholesterols, and they help to make our fatty acids that we eat more soluble, so we can destroy those with lipase, which is our next question. Lipase is controlled by a process known as interfacial activation. What that means is that these molecules really are only active where fat or lipid and water meet up. So biosalts, take our triacylglycerols that we ate, help to make them more soluble, which helps to create this lipid-water interface. And you're going to have like a lot of churning too. So you're going to have like more surface area for this lipid-water interface. And lipase is just going to go to these areas and start chopping off our tails so we can digest the fats. Lipoproteins from high to low density, right in the name. I think I highlighted the wrong one for this, because it should go, yeah, it should be this one. High density, intermediate density, low density, very low density, and chylomicrons. So the names will give you the densities for these molecules. LDL, when it's taken up by the liver, what happens? So we had a slide on this that shows this happening in detail. Basically what happens though is that when the liver takes this up, you're going to bring the LDL to the ER. And if you remember, LDL has a lot of cholesterol in it. And so what it's going to do, it's actually going to downregulate HMG-CoA reductase. And what HMG-CoA reductase is basically the enzyme that controls how much cholesterol you're making yourself. Because we can make cholesterol, but if you eat a lot of cholesterol, you're going to have it in your blood. LDL is going to be the molecule that really helps to transport that. And so if your liver is receiving a lot of LDL, that means you're receiving a lot of cholesterol. So you're going to stop making cholesterol. So you're going to down control HMG coenzyme A reductase. And what LDL also can do is if you're receiving LDL, you can upregulate how much LDL you are receiving. So what will happen is if you keep getting hit with LDL in the liver, you're actually going to decrease the amount of receptors you have, because you're basically saying to the liver, we have just received a bunch of cholesterol. Here's our signals. Start reducing the amount of cholesterol that you're making. Liver, stop actually taking LDL, because the liver is kind of like the brain of metabolism, more or less. It's controlling all your hormones for metabolism for the most part, not completely, but it is one of the major control points. So if the liver's got enough signal, it doesn't need to take up anymore, and that cholesterol can go to the body and can go to all your cells. Unfortunately, what happens there is that the LDL gets stuck inside your arteries and kind of just releases the cholesterol inside your arteries. You start getting plaques, and so all the bad stuff happens. But when we were just talking about the liver, that's what's happening. So that's why it's, I guess, Q. Looks like my letterings are very interesting there. So which lipoproteins deliver cholesterol from the cells to the liver? So looks like I have HDL. that correct? HCl. One sec, I just have to remember my whole LDL, HCl, VLDL. I'm actually not sure about that off the top of my head. Because I want to say, I'll get back to everybody on that. Unless somebody has that slide up right in front of them. I'm going to take a mulligan and get back to people on that and see what it is 100%. Because I have HCl. I'm not trusting my past self anymore. So let me get back to that. And if that is an error, since I have all your Test2 saved in my computer, I can go back and retroactively change grades if need be. So don't worry, I'll figure that one out and I'll post it on Blackboard, which one it is. So which of the following sequences is correct with regards to gluconeogenesis? So this is one of our alternative pathways. So we can't go directly all the way back up to glucose. The way we do it is that we need oxaloacetate, which will go to PEP, which can then go to G6P and glucose. So we need some alternative routes to do that. So make sure you just, and I believe I said this way back in class in March as well, but generally for gluconeogenesis, you just need to remember which enzymes are different. So that's what I would focus on when I'm studying for the exams. Which are the pathways that are different? And that should be 1, 3, and 10. It's the same enzymes that are ripe for regulation and glycolysis. They are also the enzymes we need an alternative pathway for. So if you do not have PEP carboxykinase, what will happen? So PEP carboxykinase allows us to do gluconeogenesis. So if you don't have that enzyme, you can't go through gluconeogenesis. So flux will decrease through gluconeogenesis. And that is test 2. Questions about test 2 while I take a drink here. All right. So I need to pull up test 3 now. Okay, and I'm gonna pull it up off where you can't see it in case I accidentally pull up grades. All right. So test 3 was our short test. So what diet are you most likely to promote the formation of keto bodies? So that's high fat diets. Because if you have a lot of high fat, that's where you're gonna use to make energy from, and the way you really make energy from fats that can be usable by the brain is through ketones. So high fat, high protein, low carbs will give you most likely to form the ketone bodies. And like I said at the beginning, you'll notice, and of course you've noticed you've taken, but in test 3 and 4, I have a good smattering of short answers. Like I said, I think I only have one short answer, and I think I might actually change that to multiple choice. So I am going to go back to my final that I have written, and I think now that I'm looking at it, you'll have 100% multiple choice for all of the questions, since there are so many questions to help speed you up with taking this test. Right, so why do diabetics have sweet smelling breath if you're untreated? Well, the reason for that is that you're going to start making a lot of ketone bodies. The reason why you're making ketone bodies is that for some reason or another, your body is no longer knowing insulin's there. So since you don't know that insulin's there, you're forming ketone bodies, and what happens naturally without an enzyme is that these ketone bodies break down to acetone. Acetone has a sweet smell. So that's why when untreated diabetics are breathing out, they naturally have acetone, and you can smell that on their breath. So fatty acid biosynthesis, where are two carbon fragments coming from? If you want to go back to that slide, you'll see in the diagram we're always getting two carbons from malonyl coenzyme A. So fatty acid biosynthesis always starts with malonyl coenzyme A. So tricarboxylate transport system. What is the purpose for this? So we also need acetyl-CoA at there at some point. So to do this, we need to get it into the cytoplasm. We talked about this before. A lot of stuff, there is usually no good ways for most stuff to get in and out of the mitochondria. So we need some way to do it, and that's the tricarboxylate transport system. It's another way to get a molecule out of the mitochondria without directly transporting it. It's another way that I'm losing my train of thought here, but it's just a way that we can move a molecule from one side of the membrane to another without physically moving the membrane. I hope the answer to 5 is still true, even though the rest of Texas thinks we no longer need to do that as much. Anyways, so acetyl-CoA carboxylase. Which of the following is true? So acetyl-CoA carboxylase, that is like the first step of making fatty acids. And to make your fatty acid, you're gonna take in acetyl-CoA. And when you take in acetyl-CoA, you're gonna produce malonyl coenzyme A. All right, for these questions, this isn't one of these questions, but if you noticed when taking the test, there are some questions where I have please select all that would relate to the statement. And I just figured out on Blackboard how to get partial credit for those. Took me a little bit. But so for any of those questions moving forward where I have please select all, you'll get partial credit even if you don't select all the correct answers. So if I have 2 correct answers and you select 1, you'll get 50% of that question. On the other hand, if you select, let's say there's 2 correct answers, you select 1 right and 1 wrong, then it goes back to 0, but you can't have a negative score. So if you just always select all 4, that's probably not the best strategy. But if there's 3 correct answers, you select 2 of the correct one and 1 of the false one, you still get 33% of the points. So that's just something to keep in mind. So fatty acid synthesis. So it does occur in the cytosol. That's because breakdowns generally in the mitochondria or the peroxisome. So again, this is a way to separate our 2 processes. Eukaryotes happens on a giant complex that looks like a snake. Each part of the complex is more or less like a different enzyme. That's true. The growing acyl chain is carried on the acyl carrier protein instead of coenzyme A. Yeah, so we have that chain that's like a long whip, more or less, that is moving our growing fatty acid chain around. And you need 2 NADPHs per acetyl group. Yep, so all of those are true. Humans, the direct fatty acid you're always going to make is palmitate. After you make palmitate, then you can start to make different fatty acids.
I got a question for you.
Yeah.
So for the tricarboxylate transport system, whenever it comes out of the mitochondria, it's coming out as citrate, right? It's not coming out as oxaloacetate and acetyl-CoA.
Let me look that up so we can tricarboxylate. All right. Cool.
Yeah.
All right. So what's your question on this?
So pretty much on my answer, I wrote, this is a transport channel that moves citrate from the mitochondrial matrix to the cytosol, and you gave me 0 points. and I'm wondering why. And I understand like the point is to move acetyl-CoA from the matrix to the cytosol to make fatty acids, but
So I would guess my argument there is the purpose. So the purpose is not to move citrate. The purpose is to move acetyl-CoA. So that would be my technicality, but I guess I can give you some points back on that because I guess, so that's how I'm going to roll. I'll go back and give you some points, but
yeah, that's where I go back to the whole, it's a time thing and we've only got a minute. Here's your quick answer. You know what I mean?
Yeah, that's why I'm going towards multiple choice to prevent any of those.
Yeah, definitely. Thank you.
Anything else? All right. Ubiquitin. This is actually a protein I studied, not a lot, but quite a bit in grad school. That's a soft spot in my heart. So ubiquitin, as its name, it's found everywhere inside of the cell. It does a lot of stuff, but one of the things it's most known for is to mark destruction. And so the way it does that is it needs to be activated first. So you have a more or less an E1 that activates ubiquitin. From there, you transfer it to like an E2, which is a conjugating enzyme. From your E2, you can attach it to a lysine via another enzyme called E3. Then once you have one ubiquitin on, you can start linking other ubiquitins to it using the same type of process. So amine group that is removed from amino acids is through a process called transamination. So I did have a question via email of like, how much should we know about something like transamination? And I guess I'll go back to my other points for like our cycles. And since this is a part of the urea cycle, I'm probably, I say probably, even though I've written the test and I can't remember exactly what I've written on it. I'm probably not going to ask you like, what is the exact name of the enzyme that does this step in transamination? Rather, know the process. Where does transamination fit in the urea cycle? What is the purpose of transamination in the urea cycle? What happens to the nitrogen in transamination? So be sure to refresh yourself on those. So proteins and metabolism, their lifetime is usually directed to, do you need them? So protein lives can vary based on a lot of things. But if you are a protein that's part of a metabolism, you're either going to go down or go up depending on whether you're needed right now. So if you're something that's doing glycolysis and you keep getting the signal of we don't need to do glycolysis anymore, you're probably more likely to be destroyed. And really, it's kind of a balance. It's more like you're no longer going to be created, and since you're not being recreated, the copies of you are just being destroyed now and not being replaced. That's kind of how that actually works in practice. PEST. PEST stands for the amino acids, proline, glutamine, serine, and threonine. If you have a PET sequence, that means you're going to be phosphorylated and ubiquinated, which means this is a way that we can mark proteins to be more actively destroyed than others. Lysozyme, this is where a lot of your digestion happens. Why doesn't the lysozyme eat your cell if it bursts? The reason that it doesn't do that, thankfully, is that these proteins are only active under low conditions of pH. This is actually something we talked about way back in Biochem 1. Different proteins are activated under different pHs due to different protonation states of their amino acids. Lysozyme needs a very acidic condition to have the right protonation state for this protein to be active, or these proteins to be active, rather. where does this reaction take place? So this is the urea cycle, and that was in the mitochondria. I think originally I had this in the cytosol, which is incorrect. So I believe I went back and changed that for everybody. So urea cycle is controlled by what? Well, you need the enzymes, right? So, and you need the substrates for these enzymes. So based on how much substrate you have for the enzymes, you can go faster or slower. Activation of our carbonyl phosphate synthase 1, whether that's activated or that's turned off, and how much glutamate we have, because glutamate is going to be one of these amino acids that are important in urea cycle. It's bringing us NH3. Glutamate is one of your major carriers of NH3 for you inside your cells. It's actually the amino acid that you have the most of in your cells. It's one of your major salts. Which enzyme does not appear in the urea cycle? Acetyl-coenzyme A carboxylase. That is not part of the urea cycle. What molecule does not show up in the urea cycle? That is asparagine. So that was going back to looking at your urea cycle printout and removing or just figuring out which ones appear and which ones don't. GLUT4 is glucose transporter in muscles. GLUT4. Here we're getting to our big pictures now. So mammals, what is the common thing of fuel metabolism that you see over and over and over again? Pyruvate and acetyl-CoA, they keep popping up all over the place.
So I understand what you're getting at that, but wouldn't that be a substrate used in fuel metabolism, and the end result would be H2O, CO2, and you know what I'm saying? Like I think about the product of combustion reactions is CO2 and water. I'm not thinking of the methane and the oxygen involved. That's why I went with none of the above on that answer.
So I guess for that, I would say, how do I want to word that? That's thinking, I agree with you if we are talking strictly about aerobic metabolism, while I guess this is much more broad, as in when you look at what pathways, when you look at all our pathways, when you're looking at glycolysis, when you're looking at the breakdown of fatty acids, when you're looking at the breakdown of amino acids, where do all those roads converge? And we're gonna always keep converging on our two main molecules of pyruvate and acetyl-CoA. Those can be further processed through the citric acid cycle, then to go down to electrons to make oxygen and, well, make water rather. That's what you're basically making.
I see what you're saying. When I saw the none of the above on there, that was an instant, okay, well, what else could it possibly be? Where else could all these pathways lead to end up with this byproduct at the end? And that's literally one of the reasons I was kind of like, okay, why did I get that one wrong?
Oh yeah, no, I definitely understand that like the logic there is completely makes sense. I would say that your logic isn't flawed, and I guess that's one of the hamperings of doing one of these electronic tests. Because if you were to put like on a piece of paper, none of these, because eventually oxygen is going to be broken down to water, because that's basically what happens at the very end. I probably would have still given you some points for that, because I think what my feeling on that is it's a logical answer, but if you take it all the parts in a whole, it might not be the best answer.
I know what you're saying. Now, from my perspective, imagine the answer choice, none of these isn't even there. And now it makes complete sense to me, and I know which answer to choose. And that's where like we're learning as we go. Don't get me wrong, I'm not like faulting you for the test making, but at the same time, that's something that has to be considered is from my aspect, that as soon as I see the none of the above, what else could he possibly be talking about? And so that's kind of why I'm just like, can we not have any of those on the final, please?
We're gonna still have some of those. I'm not gonna promise. I'm not gonna take away the answer of none of the above.
Then we might be having another Zoom session afterwards.
Well, if you want to argue your case on a question by question basis for each time I say none of the above, I guess I'm okay with that instead of taking away none of the above.
I got you, but like it should be like a blatantly obvious type. Like I don't know. I feel what you're saying. I totally do. But to me, when I hear end product, that's end product, not main intermediates created.
So I guess kind of just as a side point of this, the reason why that was also in there as well is because there was a specific slide in which I talked about that, where I showed all the different pathways, and I was showing everything was going to pyruvate. So that's why that question.
Tracking.
My mic probably just shut off there for a second. That's why that question was there in the first place. It's because I know, and yeah.
I got you.
So let me just pull that up. Yeah, sure. So when I made that question and when I used that question, I had this thing specifically in mind where everything is either going to go to like pyruvate or acetyl-CoA at some point during their development. And you can see that sometimes, let's say we do glucose break down to pyruvate, as in this pathway. That pyruvate is not going to go to oxygen. Let's say this pyruvate is going to be made into amino acid, right? Or let's say this acetyl-CoA is going to be made to fatty acid. So that would be the argument against everything leads to oxygen. That's only one of the pathways in the citric acid cycle, compared with everything eventually is going to go through pyruvate or acetyl-CoA onto their destination to someplace else. So my ultimate ruling on it is I'll go back and since you made a compelling argument, I will meet you halfway on that.
Yes, sir. Appreciate it.
All right. So beta pancreatic cells, how do they release insulin when blood glucose levels become high? They have the enzyme glucokinase. Glucokinase is different than hexokinase. Glucokinase only works if glycolysis levels are high, that is roughly 5.5 millimolar. Once you have glucokinase turned on, all of the energy from glycolysis in beta cells, all that's going to be used for is to pump ion pumps. And when you have these ion pumps working, that's going to be a signal for insulin to be released. So that's kind of like, I don't know, I thought that's awesome when I learned about that. That is how we know that we have high glucose levels, because your beta pancreatic cells have a special enzyme that only works at a high insulin level. So here's another one of those big pictures, what happens when insulin is secreted. So if you have insulin, you want to take up glucose, you want to make glycogen, and you want to stop gluconeogenesis. Lipolysis is actually, well, lipolysis is breaking down a lipid, so you're not going to want to do that if you have insulin. Breaking down lipids is energy. Leptin means you are full. Leptin, if you don't have it like some mice, that means you're always hungry. Type 1 diabetes is if your pancreatic beta cells aren't working for some reason. So what does AMPK do? AMP-dependent protein kinase turns on when you have AMP around. If you have AMP around, that means you're low energy, so that's going to be activating metabolic pathways to make ATP, and it's going to shut off things that make things for us. So you can usually live, well, not live. So after 40 hours, your liver glycogen is gone. So the majority of the glucose in the brain is usually just used for pumps. So like a lot of the glucose that you send, a lot of the energy you send in the brain is just used to control your sodium, potassium pumps so you can move nerve impulses. What can generate glucose for us? Liver can directly generate glucose, and interestingly enough, kidney can also make glucose for us. Adipose fat, so we're going to make ketone bodies. I guess eventually those ketone bodies can be used for energy, but you're not going to directly make glucose in the fat. Muscles aren't going to do that, generally speaking. Okay, so what happens when you're starving? So if you're starving, your kidneys are going to start giving you glucose. You're going to start using ketone bodies in your brain. You're going to start releasing fatty acids. Unfortunately, the way adipose tissue works, like your body's not very good at using fatty acids. So even when you release fatty acids, you're just going to retake up, I want to say like 70% or some ridiculous high number of them back into the adipose tissue, which is one of the reasons why it takes forever to lose weight. So metabolism muscle is controlled by glucagon, insulin, and epinephrine. And so this was another one of those things that was debated to me. I believe if you look at epinephrine, epinephrine does not have a direct control on muscle, but epinephrine does affect liver, which will have a secondary effect on making glucose up, or muscles uptake glucose. So that's why epinephrine is the correct choice there as well. And if you go back on the slide for that, it actually says that too, because in the table, it will say that epinephrine, no direct effect on muscle, but then you look on the text, it will say not a direct effect, but affects it indirectly. So that's why that was that answer. And then we get some replication. So circular DNA, what are the bubbles called? They are called these theta structures. Oh, wait, that's not theta, that's phi. Cancer metabolism, how is cancer metabolism different than normal metabolism? Cancer cells don't really do the citric acid cycle because they're building stuff all the time. So they just go through glycolysis and the pentose phosphate pathway a ton. So you'll see a lot more blood vessels going to your cancer cells, but they don't really use the oxygen. They are really more interested in using the glucose. So they're kind of starving your body. So what happens if you had hexokinase instead of glucokinase? So remember, glucokinase is that enzyme that works at high levels of glucose. So if you had hexokinase instead, your liver would compete for your cells for glucose. And that's not the liver's job. The liver is really storage and to control all the other signals. You don't want to compete for your body for glucose in the liver. Signal transduction pathway, we already talked about that back in test 1. So most organisms replicate DNA in bidirectional manner from a replication origin. So you're doing it both directions. Why do you need an Okazaki fragment? Because I can only synthesize DNA with a free 3' hydroxyl. If I want to do both strands of DNA at the same time, which are running in opposite directions, one direction, I'm always going to have a 3' hydroxyl. The other one, I have to keep making this new 3' hydroxyl that's on the ligand strand. So I always needed a primer there to always have that available for me. Any questions about that before we move into our last test?
So pretty much on the metabolism in muscles is controlled hormonally by, the only reason glucagon is on there is because it indirectly affects the muscle through other systems.
Yeah, so muscle can be indirectly affected.
Okay.
But yeah, so that's why I let that one slide when I was asked about it, because yeah, it does have an indirect effect on the metabolism of glucose.
Gotcha. Okay.
Test 4. All right, so hopefully when you see test 4 questions on the final, everybody should do all right on them, because those should be relatively fresh in your mind. So I'm gonna see if I can't go through this a little bit faster, as my throat is starting to run out of steam here. So PAL1, 5' 3' exonuclease activity. That's because PAL1 removes the RNA primer. One sec, let my dog out. Oops, sorry. So why does PAL1 have very high fidelity? Because it has the error proofing 3' to 5'. So anytime you see 3' to 5' on your polymerase, that means it can check itself. It can determine, it can chop off any nucleotide that was added incorrectly. If you don't have that, that probably means you're very error prone. Okay, so PAL1, when you cleave it, you have the cleanol fragment. Cleanol fragment does not have a 5' to 3' exonuclease activity. If I add this to DNA, I should get single-stranded DNA. That's DNA B. That's what DNA B does. Where does leading ligand strand synthesis takes place in E. coli, multi-protein replosome? And while I'm looking at this, what might be a good idea is just look back at the questions and try not to look at the answers, and try to see if you can re-answer the questions, because I might have used a couple of these on the final. Free radical. Okay, so there was some contention about this as well. What I was really looking for when in your description of a free radical is the important thing that it has an unpaired electron. True, most free radicals do happen from metabolism, but just saying like oxygen or H2O2 doesn't tell me what a free radical is. You needed to say that it had an unpaired electron. What type of damage? Pyrimidine dimers, point mutations are going to be the type of damage that free radicals do. This thing, okay, another one of those questions. This molecule is kind of too big to hydrogen bond with bases. There's really no place for it to do that. If you have normal double-stranded DNA, you can't fit that like and have it hydrogen bond. This also isn't really going to do any deamination. What it can do is if I have bases like this, and imagine like here's what I'm trying to show is here's double-stranded DNA, and just imagine, you know, I move one up and one down from that, so there's still another base between here. This thing can slide in between my bases. That's called an intercalating agent. Thymine bromide, we use that in biochem lab. That's what a thymine bromide is. This will cause a bulge in your DNA, which can lead to problems. And so your distance between bases increase, and you could have an error in replication when that happens.
Is that part of like the restriction enzyme stuff or what? Is that too much? If that's too much, we can just move on.
So I'm just trying to think exactly what mechanism. So short answer, for that answer, that question specifically, I was not looking for that much detail.
No, I was just wondering because you said we used it in lab, and I was.
Oh, what do we use it in lab
Yeah.
for? Gotcha.
Or,
No, the whole reason we use ethidium bromide is to make DNA glow under UV light. Yeah, because when I'll just do this relatively quickly. When ethidium bromide is between DNA bases, it no longer rotates. If ethidium bromide is not rotating, it will more or less fluoresce under UV light. So if it's in DNA, it's no longer rotating. We can see it under UV light. If it's not in DNA, it's free to rotate. We don't see any light from it. So that's why we use it.
Okay.
So this was, I've made the announcement. This was a question I didn't give you the correct answer for. And it was just brought to my attention this morning. So everybody got a free 2% back on test 4. So the correct answer for that is up on Blackboard. Here's one of these questions I was talking about. Which of the following statements about DNA polymerase 3 is true? So anytime I ask you for multiple answers, now you'll get partial credit even if you don't answer all of the ones correctly. So Pol3 has greater accuracy than RNA polymerase. That's true. RNA polymerase is okay to make some errors. You're just making protein anyways. Pol3 is primary replicase for E. coli. Yep, it's going to be the main one. Pol1 is the minor one that only works on the primers. Pol3 does not contain 5' 3' exonuclease. Yeah, that's Pol1. Pol1 will have that 5' to 3'. To synthesize telomeres, you need telomere 8. So telomere 8 is a ribonucleoprotein made out of RNA and proteins. RAD51, what proteins associated with it and can cause the cancer if you don't have it? BRCA1 and BRCA2. Not so much a fun fact, but interesting fact, I guess. The main cases of breast cancer, about 70-ish%, is due to a loss in BRCA1, BRCA2. I want to say it's one of those over the other, but I can't remember off the top of my head. So those are very important. If anyone's thinking about any breast cancer research ever in their future, you're going to get to know BRCA1 and BRCA2 very well. Replosome of prokaryotes only can contain 1 molecule of DNA Pol1, but 2 molecules of DNA Pol3. Why? Pol1 is only needed to remove the primer, and it's only needed on 1 strand. So basically what happens is that you have Pol3 working on each strand, the leading and the lagging. And then once you get to your primer, you're going to substitute out Pol3 and bring in Pol1 and chop off the primer and put the rest of the DNA. So that's why you have 2 copies of 1 and 1 copy of Pol1. So if we need to make an AP site, what will do that? That'll be glycosylase. The way to remember that is a glycosylase will break the glycosidic bond. And when you break that glycosidic bond, you no longer have a base. That's an AP site. Cytosine deamination leads directly to uracil. One of the reasons you don't have uracil in your DNA is so we know when cytosine has been deaminated. If you had uracil in your DNA, that would be way more error prone. So we just, somewhere along the line, organisms made thymine to instead of uracil. Beta sliding clamp sticks onto the DNA and allows your polymerase to hang on for the ride, increase processivity. Holiday junction. So here's another one of those things to not worry about so much because it's a lot. I'm not going to ask you about the intricate details of holiday junction, just like how I didn't ask you for test 4. I'm not going to ask you like 100% nitty-gritty details about holiday junctions. Part of the reason is I don't think I teach that section that well because they are still confusing to me. But basically what's a holiday junction? It's a crossover point where you have 4 strands of DNA that are making kind of like an X for a homologous recombination. So antibiotics, you can form superbugs, which are immune to these antibiotics. One of the first antibiotics is penicillin. Now a lot of bacteria are immune to penicillin because they have this gene called beta-lactamase. Why would more bacteria gain beta-lactamase? If you're getting beta-lactamase in different strains of bacteria, what that means is you have some kind of transposon event happening that's able to move this gene into the various different genomes. So it's mainly due to a transposon that is moving this antibiotic around. Which DNA polymerase has the highest fidelity? That is Pol Delta. Pol Delta has very high fidelity. Why do we need primates? Primates will add that RNA primer. We need the RNA primer because we need a 3' hydroxyl. SOS genes. SOS genes are your last defense. If you're an E. coli and you're trying to replicate your genome and you can't, after about 40 minutes, you're going to turn on these SOS genes to try to find some way to get over your stalled replication. What does RecBCD do? RecBCD will create single-stranded DNA, which will allow RecA to bind. CRISPR. Why don't you chop up your own DNA if you have CRISPR? Because CRISPR only works if you have these PAM on the 3' side. So in the host organism, you always have the same sequence that are spaced between your bacteriophage sequences that you want to cut, which then these spacers don't have the PAM sequence. The thing you want to cut, the PAM sequence, is usually super generic, where like probability states you're gonna find it near the 3' site, because it's like any nucleotide and then like a GC could be a PAM motif, and you only have 4 bases, so you're gonna find that, and so you're gonna chop. But the host organism, you're never gonna have that because you always have the same sequence next to your CRISPR sites. So what helps RNA polymerase bind? That's sigma. And what is the place called where RNA synthesis starts? That's the promoter. What are the elements that can be just about anywhere in the genome that will are activators for transcription? Those are enhancers. They can be 5', 3', or you can flip it around 180 degrees and they still work. What makes rRNA? That's Pol-1. What is RNA made for? Made from DNA. In eukaryotes, all 3 RNA polymerases recognize none of these. So our polymerases are specific. They are looking for specific sites because they all have different jobs. So it would be very problematic if they could all recognize the same thing in the genome. So it's a good thing that they all recognize different things. What RNA undergoes post-transcriptional modifications? All your RNA does in some form or another. mRNA, rRNA, and tRNA all get changed. So pre-rRNAs, what happens? Well, so pre-rRNAs basically have the same things as introns, more or less. So you need to cut off those introns. They also need to be methylated, and the things that methylate them are these small nuclear RNAs or SNO RNAs. So those are the 2 answers there. So what is true about splicing? What's true is that you have a 3' OH on your exon, and this exon will attack the 5-terminal phosphate. As very loud beeping is happening.
Sure, I'm gonna have to ask you to go out there and handle business.
Yeah, I don't know if the garbage. Yeah, I don't know if the garbage people will care if I tell them to be quiet for a second.
You don't have nerd rage ability?
I don't. I'm very calm.
It's okay. We'll handle that.
Okay. Please come over to where I live and tell them to be quiet while I'm recording. All right. What is abortive initiation? Abortive initiation is basically when you can't leave the initiation site. And it's not required. I saw that answer a couple times, and abortive initiation is like not a good thing. It's just that you are binding so tight, you can't leave. So it might just take a couple times to get off the promoter before you can actually start. Okay, so what can you have in your polymerase that allows you to bind the promoter but not do RNA synthesis? So if you just have sigma and beta, sigma is going to come and find the promoter more or less, and beta is going to help clamp on. But you don't have anything else, so you're just going to like sit there clamped onto the DNA. Group 1 and group 2. So this was another confusion that I saw. So group 1 needs a free guanosine. That comes in the form of GTP, GMP, or GDP. During that process, though, you never lose a phosphate. So you never actually use energy. You just use these guanosines as a platform to do the chemistry. So all you care is that it's a guanosine. You don't care about the phosphate. So both group 1 and group 2 actually don't consume energy in how they work. The difference is that group 1 needs a free guanosine, and group 2 forms the lariat. I hope people from Texas know what a lariat is. It's basically that rope with the circle that cowboys like go round and round and round with. Group 2 uses the internal A residue, just like your introns, basically. CTD of RNA poly 2 is important for which phase of transcription. So CTD of RNA poly 2 happens a lot. What will happen is when it's dephosphorylated, that's when initiation starts. And then when you phosphorylate it, that's when elongation happens. What sits on mRNA cap? That's a 7-methylguanosine, M7G. Poly-A tail, how does that prevent mRNA from being degraded? So again, this is another one of these answers, so I hopefully multiple choice will now make sure this doesn't happen during the final. A lot of people said they help to prevent it, kind of restated the question back to me. How does it prevent RNA from being degraded? Because by adding a tail, it's not going to be degraded. The answer is when you have a poly-A tail, you get to bind a special protein. This protein helps to shield the mRNA. What do SNO RNAs do? They will methylate rRNAs. That actually goes back to a question previously we went over. Which of these statements are false? So none of these statements. So prokaryotic, you usually have factors that can work. So you can have a row factor that can help with termination. So it can be factor dependent, and that protein is going to be row. But there are also some terminations that happen due to the sequence itself. So the sequence in the DNA will cause the RNA to self-bind itself and form what's called a hairpin. When it's in that hairpin, the RNA polymerase can no longer move, and so termination is stopped there. So remember, there's 2 ways to stop termination in prokaryotes. One's using the row factor, which is actually a protein that will come and help stop it, or the DNA sequence itself causes the RNA to bind on itself and physically stop moving forward. What's the Tata box? That's where the sigma subunit binds for RNA polymerase. So if you have insertion or deletion of 1 or 2 nucleotides, what will happen? That can cause a frame shift mutation where your set of codons have now shifted if you remove 1 or 2 nucleotides. If you have a codon does not make an amino acid but stops the ribosome, what's that called? That's a stop codon. Hopefully a lot of these in our final chapters more or less review. Unfortunately, we did not have time to get to the cool stuff of how ribosomes actually work on a molecular level, but that's where all this was leading to. Triplet codon allows many amino acids to specify for more than 1 codon. That is called degenerate. And that is the end of test 4. Yeah, so those are all 4 tests. Hopefully that was a good whirlwind refresher on the material we covered. Any questions that I can, any questions anybody might have at this point?
Is there going to be a lot of questions on that last chapter that you gave us, 27.2? Like,
Not more. No, I think I only asked like 3 or 4. And hopefully, again, those questions should be relatively, not all of them, but there are some review in there. So hopefully it won't be too bad. But
okay.
yeah, only like 3 or 4.
So I'm assuming if they're all multiple choice, we should get our grade like literally as soon as we submit it, or you're going to make us wait?
You should, I think what I'm going to do is I'm not, since people have 48 hours to take it, you will not see how you've done until the last possible time that you could have taken it. So you have Thursday and Friday to take it. You will not know how you scored until Saturday.
Okay.
Yep. I just don't want, I'm just trying to think of like somebody who took it and it's like, oh, I just want that to be
I gotcha.
no influence on anyone else. I don't know how that could be, but just, yeah, I'm just going to blanket that until you should know Saturday morning when I wake up and reset it.
Okay, that's fair. I'm just kind of shocked that you made it all multiple choice. You always said that you were never going to, that we wouldn't want a test all multiple choice. So,
well, I also never wanted the case where your test is online either. So,
that's true.
And
Fair
lectures.
Because
I
go ahead.
just, oh, well, we missed Taco Tuesday yesterday. We could have had like the most legit study group yesterday on Cinco de Mayo with tacos. Like, I had this whole thing planned out for our last semester, and it's gone. That one opportunity, all messed up.
Yeah, it's a weird time we live in.
Anyways, rant over. I just
Yeah,
want to talk about
the thing about biochemistry too, as you probably noticed for the first half of it before we went online, is that it is very memorization heavy, and unfortunately that's the class more or less. So it's been a challenge of how do I gauge a student's comprehension of material in the class that's a lot of facts. So hopefully it hasn't been too much of pain.
Single person interviews.
What's that?
Single person interviews. Sit down and pick our brains for 45 minutes.
That would have been an interesting idea. Unfortunately, I didn't think of it. Maybe I'll do that in my summer class.
Oh, I saw that you have like 19 people in there. You're welcome.
And I think a lot of those are like transfer students because now there's no time. So basically, I'm getting people who could only take it like at night too.
Oh, well, I mean, I told people, I threatened them a little bit in Chem Club, so.
I appreciate it.
Yeah. You got more work to do this summer.
That's fine.
Cool. So, final questions, nonsense.
Anything else?
Oh, when's the last time we can like reach out to you like about final questions about the final? Like, when's your cutoff?
You
Friday.
can. So you can email me. I'll say for sure, if I get any question before Wednesday at midnight, I have no hesitation in answering those. You can still reach out to me on Thursday and Friday, and I will gauge it there. I'll probably answer you.
Okay.
But if I get a question that's like, this is way too specific, and I'm not saying anybody would do this. I just have to, being the one who's presenting this, I have to be aware that it is a faint possibility. So I'll use my own judgment, and I'll probably answer you. That's what I'll say about Thursday and Friday.
Okay, I'm just curious. I wasn't sure if you were just gonna be like, nope, after I start it, no one can ask questions, or if you're gonna be cool about it and just.
You have 48 hours to take it, so I have no reason not to answer questions. So it's like I see my question copy and pasted back to me. It's like, so how do I solve this? It's like, yeah, I'm not going to answer that one. But I don't think anyone's going to do that. So. Anyone else?
I think I'm good for now.
All right. So if there's nothing else, I'm going to go take a break for my throat because I've been talking for 2 hours straight, more or less.